Hi, my name is Maggie Byers, and this is my exam 2, which is talking about the motility of bacteria and how exactly they move around. So, how do bacteria move? Bacteria uses flagella or axial filaments. So, for flagella, these are like a long external, almost like tail, helping them move around, and these flagella, they are only found in Spirilla and certain cocci. So these are like the motors of the bacteria. They allow them to swim freely in the bacteria instead of just floating around. And it's really useful in watery environments where they're needing to reach nutrients or escape harmful substances. So flagella can appear in two different arrangements. They can appear in polar arrangements, which is when the flagella are on one pole of the bacteria. And then there is the monotrichous and lophotrichous. Those are the polar arrangements. And then there is the peritrichous. This is the other arrangement, and I'll go a little bit more in depth on the next slides. So, the parts of the flagellum. I'm first going to talk about what makes up the flagellum. So there's a filament, the hook, and the basal body. The flagella uses the filament. It is the helical structure, the longest part, and it's made up of mostly proteins. So this helical structure, it's the most visible part. When you look into a microscope, this makes up for 98% of the flagellum. And this filament is inserted into the hook. So the hook basically acts as a connector or hinge between the filament and the basal body. And without it, the flagellum wouldn't be able to bend or rotate as properly as it would without it. So lastly, we have the basal body, and the basal body is buried within the membranes of the cell and is like the powerhouse. So it has a stack of rings at the bottom, and this is basically the motor. So these spin and act as a motor, which is powered by protein gradients. And it basically anchors the whole structure to the cell. So together, these three parts, they make up like a propeller system for the bacteria to move around. So now I'm going to talk about the polar flagella. And this includes the monotrichous and the lophotrichous. So monotrichous, as we said, these two, they are on one end of the pole. So monotrichous, this is one single flagellum. So they alternate between runs and tumbles, and these bacteria, they move in a cycle. So they run forward when the flagellum spins one way, and then they tumble when it spins in the opposite direction. So flagellum, these are guided by chemoreceptors, which mean they sense chemical changes in the environment, and they respond either positively towards them or negatively away from them. And if the bacteria has a cluster of flagella, these are lophotrichous. So all the flagella on this one, they rotate in the same way, they spin, they make a bundle, and this pushes the cell smoother through the cell. But if the rotation changes, the bundle will break apart and it will change the direction and cause it to tumble. So a good example of this is the spirillum. So now I'm going to talk about the peritrichous flagella. This is the other arrangement where it's not on just a pole. It's actually spread all over the surface like a cell. Kind of like hairs on the cell. So when these go, they rotate counterclockwise. This bundles together and pushes the cell in a straight line. So this is called a run. And then they can switch to counterclockwise. This is when the bundle falls apart. Oh wait, I got it wrong. The clockwise rotation is when the bundle falls apart and it makes them tumble. In the counterclockwise rotation, this is when the spin is good. They go in a straight line, run towards their stimulus or away from it. And counterclockwise is the opposite. They rotate the opposite way. The bundle isn't together. It's broken apart, and it causes them to tumble. So the tumble, it helps it reorient the cell until the stimulus is detected again. So tumbling doesn't really move the cell forward, but it can change the direction. And the cell will repeat like a run and a tumble alteration until the environment becomes better. So if it finds food to move towards, or if it runs away from danger, like E. coli is a good example of a peritracheous flagella. So I'm going to talk about chemotaxis. So chemotaxis, this is when bacteria can sense and respond to a chemical surrounding them in their environment. So chemotaxis can be positive or negative. So positive chemotaxis, this is when the cell detects something good, like a nutrient or food, and it wants to go towards it, so it's attracted to it. Whereas a negative chemotaxis, this is when there's something harmful in the environment, like they don't want to go to it. A good example is phenol. They will move away from it. This is negative chemotaxis. And the substances that cause this are chemoeffectors, and they can be any type of molecules, not just food. They can be anything the cell can sense and react to. So how do flagella move in chemotaxis? And this is really important because they move using their flagella, and this is powered by protein gradients. So the flagellum is the tiny motor that pushes the cell, and it's built into the cell envelope. So we know at the structure is the basal body, and this anchors the flagellum to the cell wall and the membrane. The basal body does all the work. It is the energy source. It powers it, and it usually comes from an ion gradient, which is like protons flowing back into the cell. And so the counterclockwise rotation. For this example, we have the peritracheous, which I wanted to show because it kind of looks like their flagella is their hair, like I said before. And now when they spin counterclockwise, the hair is like blowing in the wind, going to the positive stimulus or negative, or wherever. They're going in a straight line, the wind's blowing their hair back, and everything is good. Whereas the counterclockwise, their hair goes crazy and tumbles, and everything is sort of going wrong, changing directions. So the counterclockwise direction, it pulls all the flagella together in a little tight bundle. And together they act as a propeller and push the bacterium forward into a straight line, causing a run. And this is when they spin counterclockwise. Whereas clockwise, this is what causes a tumble. So the rotation goes opposite. It makes the bundle fall apart, and all the flagellum are going in a bunch of different directions, and this causes the bacterium to lose its motion that pushed it forwards, and instead the tumble reorients the cell and it faces new directions. So bacterial movement is basically alterations between tumbles and runs, and it helps carry the cell forward, but tumbles can also help change the direction. For dealing with chemotaxis, the cell compares concentrations as it swims. So if things are improving, say like it's getting closer to food, the motor favors the counterclockwise direction because it senses that it's good and extends the runs and it reduces the tumbles. But it can also sense if the conditions get worse, so it will increase the clockwise direction and add more tumbles, trying to find a new path. So now we're going to talk about the different types of walks. So they have different patterns in which they move around, and there is a random walk. So random walk is when the runs and tumbles are alternated randomly. And if you just look at a bacteria in a plain environment, it looks like it has a random walk. And it's just like straight runs, and they're interrupted by tumbles. But when there's a gradient, like food or nutrients are present on one side, the random walk will become biased, and it will be, the movement is going towards the attractant or away from the repellent. So if it senses that it's going the right way, like towards an attractant, then it'll make the runs longer and it'll suppress the tumbles. So it doesn't want to tumble more and change direction if it knows that it's getting towards something. But if it starts sensing something negative, then it will increase the tumbles and try and find a new path. So lastly, I want to talk about phototaxis. And phototaxis is basically chemotaxis, but it has light as a stimulus instead of chemicals. So in phototaxis, the cell will use photoreceptors. So photoreceptors, these are like special pigments, and they're able to capture the light's energy. And the photoreceptors take this signal from the light and it turns it into a message. And the message is sent through something called the transduction pathway. And eventually it reaches the flagella. So this is how it gets the signal, and the cell will either swim towards the light, like positive phototaxis, or it might swim away from the light. And it also depends on the conditions. So phototaxis in archaea are very similar to chemotaxis in E. coli, because we have positive chemotaxis and positive chemotaxis and negative chemotaxis. We also have positive phototaxis and negative phototaxis. Positive goes towards the light and negative goes away from it. So in some prokaryotes, they can detect temporal changes in the light and its intensity and how it changes over time. So the only limitation on the method of phototaxis is they can't really detect where exactly it's coming from, only the direction and the intensity of the light. So they can only detect if it's increasing or decreasing and then adjust their movement accordingly. So that is my presentation on how bacteria move, and thank you for listening.